Now let us come to chloroplasts. Plastids are macroscopic organelles found in eukaryotic plant cells. The most important plastid in plant is the chloroplast, which are characterized by the presence of important green pigment called chlorophyll. They are involved in the process of photosynthesis. The shape, size, and number of chloroplasts may vary in different cells within the species. The shape of chloroplasts may be spheroid, ovoid, or discoid, but may be frequently vesicular with colorless centers. The chloroplasts have an average diameter of 4 to 6 micrometer in higher plants, but the size may vary according to sexual and genetic differences. The number of chloroplasts in higher plants are 20 to 40 per cell, but in algae like Chlamydomonas, there is only one huge chloroplast. The chloroplast has three main components: the envelope, the stroma, and the thylakoids. The chloroplasts are enclosed by a double-layered semi-permeable membrane called envelope of about 150 angstrom thick. The stroma fills the chamber enclosed by envelope and is a kind of gel fluid phase that surrounds the thylakoids. It contains chloroplast proteins, ribosomes, and DNA. 50% of chloroplast proteins are present in the stroma. Carbon dioxide fixation and synthesis of sugar, starch, fatty acids, and some proteins take place in stroma. The thylakoids are either stacked like a pile of coins forming thylakoid grana, or may be unstacked, producing stroma thylakoids. The stroma thylakoids forms a system of anastomosing tubules connecting different grana thylakoids. The thylakoids are composed of proteins and lipids, and the chlorophyll pigment is inserted in between proteins and lipid portions. The size and number of grana in chloroplasts varies in different species, but generally there are 20 to 100 grana in each chloroplast. Its granum is about 0.4 to 0.8 micrometer in diameter and about 0.3 to 1.7 micrometer in thickness. The stroma thylakoid is much thinner as compared to grana thylakoid and contains unpigmented lipoprotein and associated enzymes which are involved in secondary reactions of photosynthesis. Chlorophyll, carotenoid molecules, and a reaction center are assembled in thylakoids, forming two photosystems, 1 and 2. Each photosystem is associated with an electron transport system and with structural proteins. Lipids present 50% of thylakoid membrane, and these include those involved in photosynthesis. The membranes of thylakoid are composed of many paracrystalline particles called quantosomes, which are photosynthetic units and are aggregated together to form a larger particles of 500 into 100 angstrom. The thylakoid membrane also contains 9 nanometer pairs which are protruding from the outer surface and are considered as enzyme complex ATPs. They are known as coupling factor, very much like the F1 particles found on the inner membrane of the mitochondria. The chloroplast is the site of photosynthesis which requires specialized spatial arrangement of chlorophyll, associated pigments, and of enzyme systems for effectively trapping light energy and storing them into compounds from which other forms of energy may later be derived for the benefit of the organism concerned.